Hey everyone, hope you guys had a good spring break and a nice Easter holiday here. We are entering the 14th week of the semester, week 14. I think it's maybe our fourth week of e-learning going on here. And so right now I'm on our homepage. I want to go over a couple of things about the assignment today. So let's go to week 14 so we can look at it. Okay. If you click on the genetic case study, it should make a copy for you that has your name in parentheses. If you're having any issues, you can always just make a copy and then share it directly with me, okay? But as long as your name is in parentheses, you do not need to worry about sharing with me. What we talked about right before break was protein synthesis and how we go from DNA to mRNA and make a protein. You're going to use that information today to look at a case. You're going to be the doctor and you're going to try to diagnose what's going on with this patient named Michael. So, in the first part, you don't need to use any technology. Just by the description here, you're going to answer questions 1, 2, and 3. If you do need a review of what a genetic disorder is, there is a link there for you. Now, the second part is when we actually take and we analyze the DNA. And so what you're given here is a chromosome. Remember, the chromosome is just a long strand of DNA separated into different genes here. And so we have lots of different genes, and they use colors here to kind of show some of the different genes here. So on chromosome 11, if we were to look at it a little bit more closely at the HBB location, we would see that the DNA, normal DNA at this point would be CCT, GAG, GAG. But Michael has a T right there instead of an A. So we need to see exactly what that means. So the first thing that we need to do is transcribe. When you transcribe, all you're doing is you are taking the code, the DNA, and you are going to transcribe it into mRNA. Just remember your base pairs. A goes with, well, in this case, for DNA, it would go with T. But don't forget that RNA doesn't have a T, it uses a U in its place. So for every C you'd have a G. The C here would be another G. A T would match up with an A, and so forth. Okay, and that's how you would get the mRNA here. To translate it into the correct amino acid sequence, you're going to have to use a codon chart. There is a link for a codon chart right here. Now, to read the codon chart, you're going to have to look at your mRNA strand, okay? You want to look at your mRNA strand, and you're going to have to find what amino acid it goes with. And so if we were to look here, CCT, our mRNA, corresponding mRNA would be GGA. And we're using codons. Codons are groups of 3, okay? So we would have GGA. When you come to the amino acid sequence then, the first base was G. So you go to this row. The second one was G. So you come over to this box because it says second base in the codon right there. So we're at GG, and it was A, which corresponds to glycine. You don't need to worry about the actual full names of these. You can just use the abbreviated amino acid here, okay? Now, if you would like some more detail, if you're struggling still, I do have the resources here on how to read a codon chart. The Amoeba sisters do a really good job at explaining and breaking it down. I also included the protein synthesis, the overview, and so I would highly suggest that. And then we have readings here. So if you click on any of those links, you will be taken to the CK12 version. Oops. And you can find out more information here too. Okay, so hopefully this helps. Use those resources. Let me know if you are still stuck, and we will work on a game plan from there. Okay, take care, and I will talk to you guys later.